So, a very good evening, one and all present over here. So, today we'll be doing the week 2 class. So, let me first share my screen. Uh, just a minute. Hmm. Okay. So, today we'll be discussing about week 2 of the our class. So, the week 2 is RNA as enzyme. So, we'll be talking the first week we have talked about the RNA world hypothesis. And in the RNA world hypothesis, one of the major point to be noted was that RNA have the catalytic function. That means RNA can work as enzymes, and this is one of the key reasons. Um, just a minute. Hmm. So, RNA as enzymes. Week 2 RNA as enzymes. So, when the last week we were studying, we discussed that in the RNA world hypothesis, we know that RNA can function as enzymes as well. So, when I say that RNA can function as enzymes, that means they can have a catalytic function. If you remember from the last class, that one of the key factors or one of the key uh, uh, points that proves the RNA world hypothesis is the catalytic nature of the enzyme. So we'll be discussing more about that in this week 2 class. So in this week 2, we had 3 lectures. The first lecture was about ribozymes, the structure of ribozymes, then the comparison of ribozyme with the protein enzymes, and the different types of ribozymes that are present. Okay. So we all know that catalytic function of RNA can be studied with the help of ribozymes. Okay, catalytic function of the RNA is due to the ribozymes. The ribozymes can help in determining the catalytic function of RNA. So, if you see, the first was four more recent lines of evidence have added much breadth and depth to the plausibility of the proposal that RNA world existed. First is the discovery of catalytic RNAs. If I talk about catalytic RNA, if I talk about catalytic RNA, I will talk about ribozymes. Just a minute, I have some network issue, it seems. Fine, I think now it's fine. Okay, so here, if you see what are the possible remnants of the RNA world, that means RNA can, how we can prove that RNA world existed because of the catalytic nature of the RNA. The catalytic nature of the RNA helps in determining many of the important functions that occurs in molecular biology. For example, if you see over here that this catalytic nature helps in self splicing of the intron. So some of the self splicing introns that occur, it is because of the catalytic nature of the RNA. Then RNase P, which is also a ribozyme that cleaves the tRNA precursor. So these all 1, 2, 3, 4, 5, these all are actually nothing but different processes that occurs in your molecular biology inside a cell that exploits the catalytic function of the RNA. Okay, the first one is the self splicing of the introns. This self splicing of the introns determines is because of the catalytic function of the RNA. The RNase P, which is a kind of a ribozyme, a type of ribozyme, it helps in cleaving the tRNA precursors and helps in the maturation of the tRNA. This also exploits the catalytic function of the ribozyme. Then the peptidyl transferase activity that happens in the ribosome during translation. Okay, during translation, the peptidyl transferase activity that happens, that is also because of the catalytic nature of the ribozymes. Then the nucleotides which are involved in metabolism signaling the self-cleaving viral RNA. So all of these phenomena are actually utilizing the catalytic activity of the ribozymes. Fine. So what is a ribozyme? Ribozyme is an RNA. Ribozyme is an RNA which functions as an enzyme. It's a ribonucleic acid only, but this ribonucleic acid functions as an enzyme. And important to note is that ribozyme is not a protein. Okay, ribozyme is not a protein, it is an RNA which has a catalytic function. Okay, so important. Then, if we see the structure of ribozyme, the ribozyme has a primary structure, a secondary structure, and a tertiary structure. Okay. So, primary structure has base, ribose, and phosphate. So, nitrogen is base, ribose sugar, and a phosphate group. And this makes the primary structure of the ribozyme. So, when we talk about primary structure of ribozyme, we are talking about ribonucleotides. Okay, so you should know the difference between ribonucleotide and ribonucleosides. So when I say it is a ribonucleotide, that means phosphate group is also added attached over it. When I talk about ribonucleosides, which this means that phosphate group is not attached over it. It is only base plus ribose sugar. So base plus ribose is ribonucleosides. Base, ribose, and phosphate is ribonucleotide. So this describes the primary structure of the RNA. And of if now we see the secondary structure of the ribozyme. The secondary structure of the ribozymes basically involves base pairing interactions. Whenever we talk about any kind of primary structure, remember, primary structure essentially tells about the sequence. Secondary structure may now the interaction between that sequence. Okay, so here we're talking about ribozyme. So in primary structure means ki what are the sequence which are attached in a linear fashion. This is also very important when I talk about the primary structure. Okay, so I am talking about the sequence in the linear fashion. And when I talk about secondary structure, it is not linear. Secondary structure is like a two-dimensional structure is formed. Now, if you look at the secondary structure, you see that because of the interactions, there is a three stem region which has formed. So, three stem region, if you see, this is your stem 1, this is your stem 2, and this is your stem 3. Your stem 1, if you see over here, after your stem 1, you have a uridine containing turn, which is known as a U turn. Then you have a stem 2 and stem 3. and stem 2 and stem 3 are joined with an augmenting helix. This is your augmenting helix. Okay, so here we're talking about the secondary structure. Primary structure is the sequence of the nucleotides in a linear fashion, and then there is an interaction. When the interaction happens between the sequence in a, then it results in a two-dimensional structure. That is why it is known as a secondary structure. It is a two-dimensional structure, and in this two-dimensional structure we have, because of the interaction, three stem regions. You can see stem 1, stem 2 and stem 3. Your stem 1 region, after your stem 1 region you have a U-turn which joins your stem 1 with stem 2. Then stem 2 is joined with stem 3 with the help of a augmenting helix. And then you see, this is the ribozyme side, this is the substrate side, and this is the cleavage side. Okay, so these three things are important to understand. Then we come to the tertiary structure. Now tertiary structure means the three-dimensional structure. Now this three-dimensional structure helps in stabilizing the whole RNA ribozyme molecule, and therefore it helps in serving the catalytic function. The actual function of the ribozyme is actually determined by the tertiary structure. So tertiary structure has to be well prepared and it should be well conserved. Only then the actual function of the ribozyme, which is the catalytic function, can be performed. Then, so like we started that the catalytic function of the RNA. So in this whole class, we'll only be studying about the catalytic function of the RNA molecule. That how? Just a minute. Okay. Hmm. So in this whole class, hmm. So just to quickly re rewind of what we are dealing. So we are actually talking about the catalytic function of the RNA. So in the previous week, we have studied that in during RNA world hypothesis, one of the major important factor which determines or which contributes to the RNA world is the catalytic nature of the RNA. Now, this catalytic nature of the RNA can be exploited or has been exploited in the molecular biology in these many ways. And then we started with ribozymes. What are the different structures of ribozyme? Primary structure, secondary structure, tertiary structure. And then we come to how ribozyme and protein enzymes are different with each other. So can you think of how a ribozyme can be different from a protein enzyme? Can you think? Okay, let me explain you. So I think I told in the previous class also. Anyways, I'll tell you again that, see, here it is already written that RNA contains only four nucleotides. Okay, you have only A, U, G, C, whereas protein has 20 amino acids. So what does it mean? It means that variation in the case of proteins is more as compared to the RNA. What does it mean? That, see, these 20 amino acids can, through various different permutation and combinations, can join and make different varieties of new uh, combinations. Whereas these four AUGC can also, in various permutation combinations, can join and make combinations, but here variations would be more because we have 20 different candidates to join, but here variation would be less because we have only four candidates to join. So therefore RNA contains only four nucleotide base compared to 20 amino acids which are found in proteins. Therefore there is a small repertoire of functional groups in RNA. Here, because we have 20 different amino acids, so there can be 20 different functional groups. Here only 4 are there. Then our ribozymes, basically we are talking about, we are comparing ribozymes with the protein enzymes. That why over the course of evolution, during the course of evolution, proteins took over the enzymatic activity which is needed in a cell and not the ribozymes. First is this, that there is a greater variation. Then RNA, that means the ribozymes, have a high density of negative charges. So what will happen? Okay, RNA, I mean, your ribozymes have a high density of negative charges. So what? What will be the effect of this? The effect of this would be that now, since it has negative charges, it can act only on substrate which are positively charged. Can it act on substrates which are negatively charged? No, because it will repel. So it can only act on substrates which are positively charged. Will it be attracted to substrates which are neutral? No. Whereas when we talk about proteins, protein enzymes, we have different amino acids, some of which are negatively charged, some of which are positively charged, some of which are hydrophobic, some of them are hydrophilic, some are polar, some are non-polar. So all kinds of chemical variety, all kinds of chemical variations are covered when we talk about considering proteins as enzymes. Whereas when we considered ribozymes, RNA as enzymes, since RNA has a high density of negative charges, therefore it can act upon substrates which are positively charged. This limits its functional capability or functional aspects. This is another reason. Then what is the third reason? Localization of bases in the interior of the duplex. So X amino acids are directed outwards from the polypeptide backbone, which means that RNA is not, in comparison to proteins, proteins are easier to be acted on the substrates because the amino acids are directed outwards. Whereas here, if you see the structural bases, the ribozymes may, the bases are in the interior of the duplexes. So that means it requires more energy because from the interior it has to first come to outwards and then it has to act on the substrates. Okay. Whereas here it is already present on the outside, it is already present on the outer side, so the energy which is required to kind of perform its function is less. Just a minute. So this means that it is easy for proteins to function as enzyme because it needs lesser energy also. So these are the reasons why over due course of evolution proteins were chosen as better candidates for the catalytic function or for catalyzing a biological reaction and not ribozymes. And also, yeah, ribozymes, I mean, RNAs are not that stable also. Proteins are stable. So you see, RNA also has a primary structure, secondary structure, tertiary structure, just like proteins. Proteins to have quaternary structures also. Here we're talking about only primary, secondary, and tertiary structure. So primary structure again essentially talks about the linear structure, the sequence of ATGC or AUGC in this case. But because we're talking about RNA, it's not just AUGC, it is base and ribose and phosphate. The sequence of base, ribose, and phosphate, that is ribonucleotides, in a linear fashion. And when this linear fashion sequence interact with each other and form a two-dimensional structure, it is called the secondary structure. And again, when this secondary structure re-interacts with each other, it forms a tertiary structure, a three-dimensional structure. And this three-dimensional structure actually serves the catalytic function. Now, see, the mechanism of catalysis is also very diverse. For example, metal ions, acid-base mechanism, small molecular metaboloids as a cofactor, substrate, tRNA for its assistant. So usually ribozyme combines several of these strategies. So what does it mean? That ribozymes to perform its catalytic function utilizes these mechanisms. First is metal ions or acid-base mechanism or small molecular metaboloids or substrate. It needs some kind of substrate for its assistant. So these are the some of the mechanisms which are exploited by your um, uh, uh, ribozymes for its function. So now we will see what are the different types of ribozymes. First, we talked about what are ribozymes, then how ribozymes are different from a protein enzyme, and now we're talking about the different types of ribozymes. Okay, so ribozymes can be classified into two types. First is metalloribozymes, and second is small ribozymes. Okay, now, metalloribozymes also has two subclasses. First is ribonuclease P and self-splicing introns, whereas small ribozymes has three different types. Hammerhead, hairpin, and HDV. HDV stands for hepatitis delta virus ribozyme. Okay. So now let us see what are metallozymes. So like I discussed in my previous class also, my way of teaching is that first we'll try to quickly revise of the content that was there in the previous week class. Okay, so like in the last week also I discussed the content uh, which was described in the week first, and then after quickly revising we went to questions. And I expect that you are already reading the content, you're already watching the videos, but my revision will just help you to gain the concepts in a better way, and then it will be better for your examination point of view also, and for your assignments also. And since we have 2 hours and we have ample amount of time, so why not utilize it and discuss and revise the theoretical portion also, and then of course questions are that we are going to do. So now let us see the first category of ribozyme, which is metallorybozymes, under which we'll be talking about ribonuclease P. So ribonuclease P, in short, we call it as RNase P. Now ribonuclease P are important for your examination point of view also, for this exam also. Why it is important? Because it catalyzes the site specific hydrolysis of precursor tRNA, which is essential for the formation of mature tRNA. What does it mean? It means that RNase P is important for the formation, for the maturation of tRNA. Okay, I'm just writing it here. RNase P is important for the maturation of tRNA. So tRNA का maturation, any question which comes which asks you the tRNA maturation, you have to say RNase-P. So how? Because it catalyzes site-specific hydrolysis of the precursor tRNA. So that means the precursor tRNA is cleaved, it is hydrolyzed, and then it leads to the formation of mature tRNA. And this catalytic activity depends on the presence of some divalent cations. That means RNase P requires certain divalent cations such as magnesium and manganese to perform its catalytic activity. And large ribozymes composed of both RNA and protein, however, RNA moiety alone is the catalyst. So this means this ribonuclease P is a large ribozyme. It is a kind of ribozyme. So this ribonuclease P is a large ribozyme and it has both RNA and protein. Both things are there, but the RNA moiety is only functioning as a catalyst. It is important. Why it is important? Because we know that, so it becomes important when we know the concept that all enzymes are proteins, but all proteins are not enzymes. Could you understand what I'm trying to say? All enzymes are proteins, but all proteins are not enzymes. That means all enzymes are essentially derived from proteins. But the sole function of proteins is not to catalyze a reaction or not to work as a enzymes. Protein can work, can function as enzyme, and protein has many other functions as well. So the protein part which is present in this ribonuclease P is not the enzymatic. Although protein is present, but the catalytic function is performed by the RNA moiety. Okay, I hope this point is clear. So this point will become clear only when you can understand the depth of this statement, that all enzymes essentially are proteins. All the enzymes are proteins, but all the proteins are not enzymes. Okay. Okay. Then so this is what was covered in lecture first. Then in lecture 2 we went ahead with understanding about the ribonuclease P and everything. So see, I'm not covering the from A to Z all the points that were covered in the class, because then it would be more than 2 3, it will take 2 to 3 hours, and I have to cover the syllabus and I have to deal the questions which are the most important thing. I'm trying to pick the important points and briefly uh, explaining you the gist of whatever has been taught in that lecture or in that week. Okay. Fine. So then lecture 2, we continued with what we were studying. So we were talking about metalloenzymes, right? So in metalloenzymes, we covered ribonuclease P. Next category is self-splicing introns. Self-splicing introns are what? What are introns, first of all? We should know what are introns. So in a gene, there are two regions. The first one is exons, and the second one is intron. Fine. If this is a gene, this is exon, this is exon, and this is exon. And this exon is intervened with introns. That means exon, intron, exon, intron. So now if I talk, this is 5 prime. You have 5 prime UTR also, you have 3 prime UTR also. So this means these exonic regions are intervened by certain sequences which are known as introns. And what is so special about these introns is that the coding region of the gene, that means the region of the gene which is actually responsible for carrying the genetic information from one cell to another, or from carrying the important function or role from one cell to another, are the exonic regions. So exons are the coding regions and introns are the non-coding regions. That means introns need not to be present in the mature mRNA. So these introns are spliced out. Okay? Spliced out means that these introns are cut, and when the mature mRNA is formed, you do not have these introns. You have only exons E1, E2, A3, and many more. So this means these introns have to be spliced out. So the splicing of the intron can happen in many ways. Fine. This splicing can happen in such a way that some complex, which is known as a spliceosome complex, some external spliceosome complex is working on these introns to remove it. Okay. Some some external, not external as in not the extrinsic nature, not the intrinsic nature of the intron, because of some extrinsic factors from somewhere else, spliceosome complex is coming and it is acting on this intron so that these introns can be removed or spliced out, or these introns themselves have the property, or these introns themselves have a capability to get spliced out. You understand? So if these introns themselves have a capability to splice out, that means these are the self-splicing introns. And these self-splicing introns are a kind of metalloribozymes. That means they are a kind of ribozymes. That means they are a kind of RNA. Obviously, they are RNA, which has a catalytic function. So catalytic function means what? That means they have the ability to hydrolyze its bonds and just come out of the sequence. So this is, and why it is under metalloribozymes, because these metalloribozymes ka definition is that these ribozymes depends on the presence of some metal ions. Okay, so the catalytic activity of these ribozymes is dependent on the presence of some cations some ions. That is why they are metallorybozymes. Some metal ions are needed. Like here, magnesium and manganese is needed. So here also some divalent cations are needed for its function. So that is why they are under the category of metallorybozymes, because for their catalytic activity you need some metal ions. So here these self-splicing introns are actually large introns that are able to splice out themselves. Okay, and in bacteria as well as eukaryotes, pre-RNA of protosome, tetrahymena, primary transcripts of the mitochondria. This is just an example. So now you see what is the difference between a normal splicing and when we talk about self-splicing. Unlike common introns, self-splicing intron can splice themselves out of the pre-mRNA without the need of spliceosome. So if you know the process of transcription, which I assume that you have a bit basic knowledge, so after transcription, your mRNA is formed from DNA. Okay, just to make things a little more clear, this is your DNA. From DNA, your mRNA is formed. Then your proteins are formed. And this is known as central dogma of life. This is your central dogma. DNA to DNA, if it is formed, it is called as replication. DNA to RNA, it is called as transcription. And RNA to protein, translation. So when this DNA to I'm here I'm talking about mRNA only, but DNA to RNA is transcription. So this RNA when it is formed, this RNA when it is formed, it is a primary mRNA. And there are some kind of post processing that happens that converts this primary mRNA into a mature mRNA. And when I talk about some kinds of post processing, then what are these? That is 5' capping, 3' poly A tail, and splicing. After these three, you have the mature mRNA. For this splicing to take place, you have a, just check the spelling, spliceosome complex. So what happens that this, I'm not sure, I mean, this is not the spelling I know. Splice, so what, just a minute. What is the spelling of spliceosome? Oh, I was writing correct only. This spliceosome complex helps in the removal of introns. This is a general phenomena that happens. Generally, you have a spliceosome complex which helps in the removal of the introns. But if your certain introns are self-splicing introns, which are actually your ribozymes. So these introns does not require this spliceosome complex. Okay. So that is what is the difference between a self-splicing and a normal splicing. In normal splicing, spliceosome complex is needed, whereas in self-splicing, spliceosome complex is not needed. Okay. Then what is the next point? Hmm. Although self-splicing introns can remove themselves from RNA in the absence of any protein in vitro, in many cases, self-splicing proceeds in the presence of certain proteins increase the efficiency of splicing. That means that um, This is spliceosome, which is required for normal splicing. But these self-splicing introns, they do not require any kind of protein or any complex for its function to be performed. However, if you externally provide some kind of a proteins, it can just increase the efficiency of splicing. That means it has the ability to perform the task itself, but if you're providing some kind of protein, it is increasing the efficiency. Whereas in the normal case of splicing, it has no ability to perform it. It is mandatory to provide spliceosome or any kind of protein complex. Here it is not mandatory, it can perform, but if you provide some kind of proteins in vivo, if certain proteins are present, then it is the efficiency is increased. Then self splicing introns mediate only one round of RNA processing, unlike protein enzymes. So this is a problem, that self splicing introns only mediate one round of RNA processing. Okay, so now comes small ribozymes. So looking at the category where it was, yeah. So ribozymes are of many types. The first category is the metalloribozymes, in which there is ribonuclease P, and second is self-splicing introns. Ribonuclease P, in short, we call it as RNase P. It is required for the maturation of the tRNA. So how? Because it catalyzes the site-specific hydrolysis of the precursor tRNA, which is essential for the formation of mature tRNA. And the catalytic activity of this RNase P depends on divalent cations such as magnesium and manganese. Then we come to the self-splicing introns. In self-splicing intron, what happens? Self-splicing introns have the capability to get spliced out by themselves. However, if any external protein complex is provided to them, the efficiency of this splicing is increased. How the efficiency of splicing is increased? Because this will stabilize the correct structure of RNA. That means uh, the structure of RNA is stabilized. And but there is a problem that these self-splicing introns can mediate only one round of RNA processing. Next, we come to the small ribozymes. In small ribozymes, we have three subcategories: hammerhead ribozymes, hairpin ribozymes, and HDV, that is hepatitis delta virus ribozymes. So let us see one by one what are these small ribozymes. And after this, uh, just a minute. Mm. So it seems that you have two TAs for this course, and both of them had class at the same time. Like I got to know, both of them had the classes from 7 to 9. So it's actually difficult for you because it's your loss, because you can attend only one of the classes. So I mean, if you can talk to the NPTEL support team, you can just tell them, because it is not beneficial for you, right? Because if you want to attend both the classes, you cannot do it because both the classes are at the same time. I will also try to talk to them that if the classes are at the same time, it's difficult for the students to attend both of them. So it's a loss for you only. Anyways, so mm, yeah, so now let us move to the category of small ribozymes. So small ribozymes have hammerhead ribozymes, hairpin ribozymes, and HDV, which is hepatitis delta virus ribozymes. Hmm. Small ribozymes of viroids and satellites. What does it mean? We have satellite viruses. What are satellite viruses? These are small RNA viruses or RNA molecules, and their multiplication depends on the mechanism of a host cell and on the co-infection of a host cell with a helper virus. So these are small ribozymes which are basically involved in your viroids and satellites. So ribozyme is a part of a larger RNA, that is viral satellite, that is being replicated by host RNA polymerase. Okay, this is not much needed. So now let us see what are hairpin ribozymes. So basically, when we are talking about small ribozymes, these are your uh, satellite RNAs only. Satellite RNA means helper. These are the helper virus ka RNA. So when we talk about hairpin ribozymes, these are plant virus. Let me just increase it for you. Yeah. So when I talk about hairpin ribozymes, these are plant virus satellite RNA which mediate rolling circle replication. So hairpin ribozymes are plant virus satellite RNAs. This is what is important. These are plant virus satellite RNAs. Then the lecture 3, it was all continued where where you were taught about the hammerhead ribozymes, hepatitis delta virus, and riboswitches. So hammerhead ribozymes, so what, these are just the screenshot of your own lectures. Okay, I have just taken these screenshot for you so that there is some content for me to explain you, and I don't want to explain content which is out of your syllabus. I want to explain you the content which is very much present in your syllabus, then only it is beneficial for you, right? So I have taken these screenshots from the course itself so that I can explain you. So hammerhead ribozymes, so this is also an RNA molecule, but it catalyzes cleavage and joining uh, at specific site within the RNA molecule. So basically, this is used for some kind of targeted RNA cleavage experiments. And uh, if you want to understand this, so suppose you have your RNA. Within this RNA, if you want to cut at a point, this has to be a very specific cut. So in such kind of experiments, hammerhead ribozymes can be used. And because it catalyzes the cleavage and joining, reversible cleaving means it can be reverted back. It catalyzes the cleavage and joining. This particular property has been exploited in some kind of therapeutic applications. Okay. Then hammerhead ribozymes RNA were originally discovered in two classes of plant virus like RNAs, satellite RNAs and viroids, and they have been found to be widely dispersed within many forms of life like insects, plants and mammals. Not very much to explain. Then hepatitis delta virus. Again, hepatitis delta virus is a very well known RNA satellite virus of hepatitis B virus. So, can anybody tell me what is the meaning of a satellite virus? When I talk about satellite virus, what is the meaning of it? Satellite virus. Anybody knows? Hmm? What is a satellite virus? Can anybody tell me? Because for when I'm talking about small RNA, satellite virus is important, becomes important point to understand. Satellite virus is nothing but uh, it's a viral agent that cannot replicate on its own. So when I talk about satellite virus, important criteria of satellite virus is that it cannot replicate on its own and it requires a helper virus to replicate. Okay, so that satellite viruses cannot replicate on its own and it requires a helper virus to replicate. And why it is so? Because the genes which are needed for the replications are not present in the satellite viruses. So this HDV is also a satellite virus of hepatitis B virus. That means this hepatitis delta virus is requires a helper virus which is hepatitis B, HBV, and this HDV is a ribozyme. Basically, it's a non-coding RNA, and it is thought to be the only catalytic RNA which is required for the viability of a human pathogen. That is, human pathogen is affected only by this is only known catalytic RNA. And the ribozyme acts to process the RNA transcript to unit length by a self cleavage reactions. And this is all easy. Just to read, I mean, nothing much to explain over here. Then riboswitches. So now, what are riboswitches? Hmm, just a minute. Yeah. So can anybody tell me what are riboswitches? Anybody has any idea? So riboswitches are the RNA segments that regulate gene expression by binding to small molecules. Okay. So basically, they control the gene expression by binding to other small molecules. And these small molecules can be either coenzymes or amino acids or nucleobases. These small molecules can be anything. So riboswitches switches are the bacterial mRNA that control gene expression via binding of small molecules. And basically they are found in the untranslated regions of the mRNA. So you know what are untranslated regions? Untranslated regions are the regions which are not the intronic regions but which are not translated. I mean, in the translation, you will not see those untranslated regions. So if this is your mRNA, you have a 5 prime UTR region and you have a 3 prime UTR region. This means that this region is not translated into protein and this region is also not translated into proteins, but these are not the intronic regions. But they are not translated. So these riboswitches are found in the untranslated regions of the mRNA. And when riboswitches bind to the target molecule, it kind of changes its shape, and this change in shape helps in the activation or repression of its downstream genes. Okay, so what happens that you have riboswitches are those RNA segments, small RNA segments that will bind to either amino acids or coenzymes or nucleobases. When they bind to their substrate molecules, it kind of changes its shape. And when it changes its shape, then this change in shape activates or represses the downstream genes. So this is how your riboswitches works. Okay. So now we'll try to solve some questions. After solving, so I have already covered what was all taught in this in week 2 lecture. After covering questions, I'll just quickly revise it. So for the questions, I'll give 2 minutes, 2 minutes for you to think, and you can write it in the message box, and then we'll try to see the answers. So the first question is, which of these do not represents ribozymes? Hepatitis delta virus, ribonuclease P, ribosomes, or RNA loop? It is 7:49. I will switch the screen at 8:02. 2 minutes I give for each question. I'll just put it like this. Hmm? Okay, there's some. Hmm. So, what is the answer of this question? You can type it in the message box if you know the answer of this question. Question is, which of these do not represents ribozymes? Hepatitis delta virus, ribonuclease P, ribosomes, and RNA loop. Hmm. Okay, let me see. Why no one is answering? You're just leaving, you don't want to answer. Anyways, so the correct answer is, I will give you 2 minutes. It's up to you whether you want to use those 2 minutes to answer or not. But it would be better if you try to answer something on the message box. It will make this class interactive. It's good for you only. Anyways, so the correct answer is RNA loop. So RNA loop is not a ribozyme. Rest all are ribozymes. So we have already studied that hepatitis delta virus, ribonuclease P, ribosomes, these all are ribozymes. See here, when we talked about what are the different categories of ribozymes. So ribozymes mein there are two categories, metalloribozymes and small ribozymes. In metalloribozymes you have ribonuclease P. So that means ribonuclease P is a ribozyme. Small ribozyme may we have HDV. And ribosomes also are have ribozymes. Why? Because ribosomes perform the peptidyl transferase activity, and this peptidyl transferase activity of the ribosome is because of the RNA, which is actually a ribozyme. I mean the catalytic RNA. Ribozyme is nothing but a catalytic RNA. Okay. So which means we are left with only one option, which is RNA loop. So the correct answer for this question is RNA loop. And also, I mean, if you try to see here, just a minute. Yeah. RNA loop is basically a three-stranded nucleic acid which has a DNA RNA hybrid and a displaced DNA strand. So it is nowhere it has any catalytic function. Okay. So when I talk about RNA loop, so RNA loop is nothing but a three-stranded nucleic acid structure. Okay. It's a three-stranded nucleic acid structure which has a DNA RNA hybrid and a displaced DNA strand. Fine. Now, next question is, ribozyme contain cofactors which are important for A. RNA primary structure, B. RNA secondary structure, C. Structural diversity, or D. RNA protein interaction. What is the correct answer? I give you 2 minutes. Then you try to answer it. It's 7:54 right now. I will switch the screen at 7:56. Okay. Think and try to answer. One minute more. Think what can be the answer. Okay, I'm switching the screen now. Fine. The correct answer for this question is RNA secondary structure. So, ribozyme contain cofactors which are important for, we need to know that whether they are important for the primary structure of RNA, for the secondary structure of RNA, or for the structural diversity, or for the RNA protein interaction. This was the question, right? So the correct answer is RNA secondary structure. Why? Because ribozymes are RNA molecules with catalytic activity. These RNA molecules have catalytic activity, I told you, and they often require cofactors such as metal ions to stabilize their secondary structures, which is crucial for the proper folding and catalytic function. Important is that these metal ions, which serves as cofactors in many cases, are crucial. They are crucial for the proper folding of the RNA molecule. Okay, and this ultimately helps in the catalytic function. See here, secondary structure. Hmm. Your secondary structure requires some kinds of cofactors for its stabilization. And when these cofactors are, just a minute. Hmm. And when these cofactors comes and join, then it stabilizes your secondary structure. Okay. So I hope this question is clear to everybody. Okay. Next question is RNA sp is important for A. RNA stabilization, B. Pre-tRNA maturation, C. Ribozyme action, and D. Nitrogen base modification. What is the correct answer? After 2 minutes, I'll switch the screen and I'll tell the correct answer. 2 minutes for you to think. If you know the answer, you type it in the box. Otherwise, I'm anyways going to explain to you. We have already talked about what is RNAsP. We've already talked about where RNAsP is needed and which category this RNAsP belongs to. So you should be able to answer this one. Okay. It's 7:59 right now. I'll switch at 8 2. 1 minute more, then I'll switch the screen. Okay. Let us try to see what is the correct answer. Mhm. There are many people. Okay, fine. So, the correct answer to this question is pre-tRNA maturation. Okay, so RNA-sp. What is RNA-sp? Let us see. We have already discussed it. So, RNA-sp is a kind of ribozyme which belongs to the category of metalloribozymes. We have already discussed that ribozymes have two categories. Okay. What are the two categories of ribozymes? The two categories of ribozymes are metallorybozymes and small ribozymes. Under metallorybozymes, you have ribonuclease P and self splicing introns. So now this ribonuclease P is important. I have already told you, I'm repeating it once again, that these ribonuclease P helps in catalyzing the site specific hydrolysis of precursor tRNA. That means that these RNAs P help in the maturation of tRNA. And how they mature your tRNA? By cleaving, by the hydrolysis of precursor tRNA. That means if you look at this question, yeah, that ribonuclease P is important for RNA stabilization, ribozyme action, nitrogen base modification, or pre tRNA maturation. The correct answer is pre tRNA maturation. Okay, next question is, so for the people who have just joined, we have first discussed about whatever has been taught in the previous weeks. I mean, in week 2, we have very briefly revised all the contents of the lecture 1, lecture 2, and lecture 3, and then we have moved on to the questions. So now we are dealing with the questions. If you know the answer, after each question, I give you 2 minutes. If you know the answer, you can type it in the message box. Even if you don't know, you can just try to attempt it. We'll see whether we are able to get your concept clear or not. Anyways, after every question, I'll try to explain the concept which is behind that question. But it is important for me to for me to you guys to interact, because when you will interact, then only it will be like a interactive and a discussion kind of a session. Fine. So this question is, during protein synthesis in ribosomes, peptide bond formation is by protein, tRNA, rRNA, or mRNA. Okay. Right now it is 8:03. I will switch the screen at 8:05. Okay. You just think what can be the answer and try to type it in the message box. I'll see whether you are able to follow it up or not. Anyways, I'm going to explain you. Try to look at the question. Question is saying peptide bond formation. So peptide bond formation, if you know the different types of RNA, you have mRNA, rRNA, and tRNA. In which of the different type of RNA peptide bond formation happens? I mean, which subtype of RNA helps in protein synthesis? Because peptide bond formation happens during protein synthesis, right? 1 minute more. Okay, let me switch the screen now. Why nobody is trying to answer? Nobody knows the answer of this question? Okay, anyways. So the correct answer is RNA. Why? See, during protein synthesis in ribosomes, peptide bond formation is by. So when they're talking about peptide bond formation, when peptide bond formation occurs, peptide bonds are present in which? molecules. They are present in proteins. So when I'm talking about peptide bond formation, that means that I'm talking about the formation of proteins. So formation of proteins means which process am I talking about? Or are they talking about? They're talking about translation. When they're talking about peptide bond formation, that means they're talking about translation. So translation, now we have three different types of RNAs. mRNA, rRNA, and tRNA. mRNA, rRNA, and tRNA. All of these three RNA are involved in peptide bond, in protein synthesis. But the function of each and every different type of RNA is different. mRNA provides the template, tRNA brings the amino acid, and rRNA helps in the formation of peptide bonds. So tRNA cannot be the option, mRNA cannot be the option, protein protein synthesis is only happening, so this cannot be the option. So the correct option would be RNA. It should actually be rRNA, which is ribosomal RNA, but here you have to choose the best amongst the options that are provided to you. That is why RNA will be the correct answer, because mRNA is not going to help in peptide bond formation. mRNA is the template which is taken, and accordingly the amino acids are incorporated, okay, in the triplets, the codons. And tRNA brings the charged amino acid. So tRNA cannot be the option, protein cannot be the option, because we are talking about protein synthesis. So what is left with us? RNA. So RNA, sometimes what happens when you are solving MCQs, okay, this is a trick for you to understand. Even when you're going to compete for some competitive examination, you might have options in which you will feel that none of the options is 100% correct. Then you have to use the option which is comparatively the most correct amongst the options given to you. For example, here RNA is not the correct answer actually, because RNA can be anything. It can be tRNA, it can be mRNA, it can be rRNA. But amongst the options provided, which is the best suitable, that we have to select. So accordingly, RNA becomes the correct answer. Fine. Now let us move to the next question. Easy. This, so I think you should be able to answer. I'll be very happy if you're answering it. DNA to RNA to protein connection is called DNA dogma, central dogma, protein dogma, or ribosome dogma. Right now it is 8:09. I will switch at 8:11. 1 minute more. Okay, now I'm switching the screen. It is an easy one only. Let us see. Nobody is again answering. Why you are not answering? Asiba, Anubu, you were answering the last class, right? Why are you not attempting? This is easy one now. Have you studied about the central dogma of life? Hmm? Just a minute. Somebody wants to join. Okay, fine. Let us see. So the central DNA to RNA and to protein connection is known as central dogma. Okay, this is your the correct answer is central dogma. So DNA to DNA, if see this is DNA to RNA to protein. If it is connecting it, if it is DNA to DNA, it is replication. DNA to RNA, transcription. Okay. RNA to DNA, it is reverse transcription. Reverse. And RNA to protein, translation. Okay. This is your central dogma. Fine. Next question is, which is not a ribozyme? RNA is P, hammerhead, HDV, and HIV. Which is not a ribozyme? Think all this time we have been discussing about RNAsP. Also, we have discussed about hammerheads. HDB also we have discussed. I've already told you the answer now. Anyways, I'll give you 2 minutes. Right now it is 8:13. I'll switch the screen at 8:15 and then we'll see. Okay? Try to answer this one. 1 minute more left. Hmm. Think. Okay. Okay, I'm switching the screen now. Oh, good. Now I think many have answered. Asiba has answered HIV, Anubu has answered HIV, Jency has answered HIV, and Fabina has also answered HIV. Very good. So it is the correct answer. HIV is the correct answer. So RNase P, it is a kind of a metalloribozyme, and RNase P is required for the maturation of tRNA. Hammerhead and HDV, hepatitis delta virus, these are the small ribozymes category, and these are satellite viruses. So this is your, so what is left with us is HIV. HIV is not a ribozyme. Then next question is easy ones only. Every living bacteria contain ribozymes. True or false? 8:15. I'll switch at 8:17. Easy only, but chalo. Every living bacteria contain ribozymes. You have 1 minute more. Okay, I'm switching the screen now. Okay. So Asiba has answered two true. Jency has answered true. Anubu has answered true, and Fabina has also answered true. So it is correct. So every living bacteria contain ribozymes. It is true that ribozymes are present in every living bacteria in some or the other form, because wherever you have protein synthesis, ribozymes are present. Wherever you have RNA, then obviously some catalytic function is there, ribozymes are present. See, ribozymes are what? They are nothing but just the RNA which has a catalytic function. And RNA is present in living cells, in living bacterial system. So ribozymes are also there. Okay. Next is RNA and DNA are same at molecular level. True or false? Right now it is 8:18. I will switch the screen after 2 minutes. That is at 8:20 I will switch the screen. RNA and DNA are same at molecular level. True or false? 1 minute more. Okay, now I'm switching the screen. Hmm. Let us see. Okay. Asiba has answered false. Anubu has answered false. Jinsi has answered false. And Roshan has also answered false. And yes, it is correct that the correct answer is false. How it is? See, when we're talking about DNA and RNA at the molecular level, then DNA is ATGC. Okay. And RNA is also ATGC? No. RNA is AUGC. So there only first difference comes. First thing. Second thing is that ATGC, AUGC is the first difference. Second is that DNA is deoxyribonucleic acid, whereas RNA is ribonucleic acid. That means deoxy. That means at the second carbon, there is only H. Whereas at the second carbon, in the case of RNA, there is OH. So that is your second difference. Okay. That is the second difference. So, and these differences have a great impact on the stability of DNA and RNA. Why? Because RNA becomes less stable at the 2' carbon because of the 2' H 2' OH which is present. It becomes less stable. Whereas the 2' H which is present in the DNA, it makes it more stable. Okay. So this is the difference. Fine. Good that you are answering now. It's very good to see. Now, next question is RNA secondary structure vary based on base sequence. True or false? It's 8:23. I will switch the screen at 8:25. Okay. Try to answer this question. One more minute left. Okay. Shall I switch the screen now? Hmm. Let us see. Uh, Roshan has answered true. Anubu has answered true. I don't know whether this is for this question or I think this question only, no? So Jansi has answered true. Fabina has answered true. Then who else? Anubu has answered true and Roshan has answered true. Okay. So RNA secondary structure vary based on base sequence. Obviously, it will vary on the basis of the base sequence. Why? Because secondary structure is because of the interactive forces that happens between the primary structure, and the kind of interactive forces that happens between the primary structure is only going to determine what kind of secondary structures are going to find. So the attractive forces will depends on what? It depends on the this thing sequence. The attractive forces depends on the sequence. So if the sequence is different, then the attractive forces will be different, and then the secondary structure will be also different. Also, the secondary structure of RNA includes what kind of structures? Hairpins, loops, bulges, stems. And these structures like hairpins, loops, bulges, and stems, they are highly dependent on the base sequence. The specific pattern of complementary base pairing between EU, AU, and GC, this determines how the RNA strand folds into its secondary structure. And variation in the nucleotide sequence, it will lead to different pairing possibilities, and therefore the overall structure is changed. So when I talk about RNA ka secondary structures of RNA, you have hairpin loop, okay. Then what kind of structures you have? You have stems, you have bulges. These are some of the secondary structures which are present. So these secondary structures will depend on the base sequence of the primary structure. And base, if the base sequence is different, the arrangement of the secondary structures will be different. So the correct answer is true. Okay. Next question, which is probably the last question. Metal ions contribute to ribozyme structure. True or false? Right now it is 8:29. I will switch the screen at 8:31. Okay. One more minute left. Metal ions contribute to ribosome structure. True or false? Hmm? I'll switch the screen after 1 minute. Okay, I'm switching the screen now. Hmm. So let's see what you have answered. Jency has answered true. Asiba has answered true. Anubu has answered true. Fabina has answered true. And Roshan has also answered true. That means metal ions contribute to ribosome structure. True. And this is the correct answer. So how it is true? So we know that ribozymes can be of several types. Okay. Two things are there to understand when we talk about that metal ions contribute to ribozyme structure. First thing is that we know that ribozyme has a primary structure, a secondary structure, and a tertiary structure. We have already discussed it. Okay, just a minute. Yeah. So we know that a ribozyme has a primary structure, a secondary structure, and tertiary structure. In the secondary structure, the stabilization of the secondary structure is very much dependent on the presence of certain kinds of divalent cations. Magnesium, manganese, some cations are needed for the stabilization of secondary structure. So here itself we know that the secondary structure for secondary structure stabilization, metal ions are needed. Then one proof, second number of proof is that we have a category of ribozymes which are known as metalloribozymes. Okay, so this category itself suggests that metalloribozymes are those ribozymes which require certain metal ions for their catalytic function to happen. Okay, for example, you talk about ribonuclease P, which is known as RNase P. So this RNase P requires the activity of divalent cations such as magnesium and manganese for its function. So this is again second number proof that ribozymes require metal ions for its function. Okay, so uh, just a minute. Yeah, so let us quickly, very quickly, maybe in just 5 6 minutes, we'll try to summarize whatever we have studied today. Okay, so RNAs are enzymes. So yesterday, so in the last class when we were talking about the RNA world hypothesis. In the RNA world hypothesis, we talked three major characteristics which proves that RNA can function as enzymes or RNA existed. First was that RNA is self-replicatory. Second was that, and one of the important point was that RNA has a catalytic activity, the catalytic activity of the RNA, and then RNA could carry genetic information. These were the three things: self replication, genetic information carrier, and catalytic nature. So, in this week, we have studied about the property number 2, that is the catalytic nature of the RNA. The catalytic nature of the RNA or the RNA which have the catalytic property are known as ribozymes, and these ribozymes are nothing but your RNA which has an enzymatic feature or a catalytic features. These are not proteins. These ribozymes have a primary structure, secondary structure, and tertiary structure. Primary structure is the sequence of AUGC plus ribose and phosphate. That means primary structure is the sequence of your ribonucleotides. Just a minute. Hmm. Hmm. So primary structure is the sequence of your ribonucleo. Uh-huh. Hmm. Primary structure is the sequence of your ribonucleotides. This primary structure then forms the secondary structure, and then secondary structure ultimately coils to form the tertiary structure, and this tertiary structure is important for serving the catalytic function. Then we see that how ribozyme can be different from protein, because RNA has only 4 nucleotide bases AUGC, whereas proteins have approximately 20 amino acids. So there are more number of individual mole substrates. Therefore, there is a greater variety that proteins can provide in comparison to your ribozymes. Then, ribozymes have a high density of negative charges. That means, since ribozyme has a high density of negative charges, it can only go and act on the substrates which are positively charged. Whereas proteins are negative. We have amino acids which are negatively charged amino acids also, positively charged amino acid also, hydrophilic also, hydrophobic also, polar also, non-polar also. Different kinds of amino acids are there. That means it can act on a greater array of substrates as compared to ribozymes. Then in the case of ribozymes, your bases are localized in the interior, whereas in the case of proteins, amino acids are actually directed outward. So when the bases are interior, more energy is needed, because first the bases have to come outside and then only it can act over the substrates. Whereas here in the case of amino proteins working as enzymes, amino acids are already directed outwards, so lesser energy is needed. So therefore these are the basic different comparison between a ribozyme which And I mean RNA working as a enzyme or a protein working as an enzyme. Then we studied that there are different categories of ribozymes which are present. The first one is the metalloриbozymes and second one are the small ribozymes. Metalloриbozymes have ribonuclease P and self-splicing introns, and small ribozymes have hammerhead ribozymes, hairpin ribozymes, and HDV hepatitis delta virus ribozymes. RNase P is required for the maturation of the tRNA. Self-splicing introns. Self-splicing introns are the introns which are able to splice by themselves. So there are introns. We know that splicing is a post-transcriptional modification. What is a post-transcriptional modification? That after transcription, this modification happens for the conversion of a primary mRNA into a mature mRNA. Okay, and this post-transcriptional modification requires the removal of the introns. So these introns can be removed either with the help of a spliceosome complex, or some introns have their intrinsic capability to get removed by themselves, and those introns are known as self-splicing introns which can be removed by themselves. However, if we provide any external proteins or external complex to these self-splicing introns also, then the efficiency of splicing is increased. I mean, they can function by their own, but the efficiency is increased if you're providing a protein. But the again, a point is that self-splicing introns can mediate only one round of RNA processing. It cannot do more than that. So this is your self-splicing introns. Then the next category was small ribozymes. So under small ribozymes, we have hammerhead, hairpin, and HDV, which is hepatitis delta virus. So hairpin ribozymes are basically plant virus satellite RNA which mediates the rolling circle replication. So these ribozymes helps in the rolling circle replication. Hairpin ribozymes are the plant virus which helps in the rolling circle mechanism. Just a minute. Then yeah, then we have hammerhead ribozymes and hepatitis delta ribozymes. So hammerhead ribozymes, again, it is a ribozyme which it responsible for the targeted cleavage and joining within the RNA molecule. So because it can cut and join in a reversible fashion in an RNA molecule, therefore they are used in various kinds of therapeutic studies. It has a lot of therapeutic applications. Then we have HDV, which is hepatitis delta virus. So hepatitis delta virus is an RNA satellite virus of hepatitis B virus. Okay, and this HDV is a non-coding RNA, and it is the only catalytic RNA. This is important. It is the only catalytic RNA which is known to be required for the viability of human pathogens. So and then we have riboswitches. So riboswitches, what are riboswitches? Riboswitches are nothing but those RNA, those ribozymes, those RNA molecules which join to certain or which bind to certain small molecules such as coenzymes, amino acids, nucleobases. And after the binding to these small molecules, what happens? These riboswitches change their shape. Okay, so these ribose switches are the mRNA portions, are the RNA portions which have a catalytic function. They go and they bind to some small molecules, and when they bind to those small molecules, they changes its shape. And when it changes its shape, then it causes the activation or repression of the downstream target genes. So basically, these ribose switches are helping in the either in the activation or in the repression of the downstream genes. And how it is doing so? It is doing so by binding to some small molecules and changing its shape afterwards. So this is what ribose switches is about. So your essentially dealt week 2 essentially dealt with the on focusing on the catalytic property of the RNA. The catalytic property of the RNA, that is catalytic RNA, is known as ribozymes. What are ribozymes? What are the structures of ribozymes? What are the different types of ribozymes, and what are the functions of different types of ribozymes? But since it's an RNA biology course, I mean, it's just the starting of the course, anyways. There are two things that are very much important for you to understand. I mean, actually, there are, yeah, two things. Actually, there are three. First is the process of transcription, how transcription happens, what are the different kinds of post transcriptional modifications, and also you should know translation. because translation में ही your ribozyme वाला property is more used. Okay, we talk about ribosomes. Ribosomes also have a ribozyme property, the peptidyl transferase activity, which happens during peptide bond formation. That is nothing but ribozymes, right? So there also ribozyme property is needed. So therefore transcription and translation both are needed when we are studying about in this course. So transcription, post transcriptional modifications, and translation, all these three properties are needed in this course for you to study. So this in this class we have studied about the catalytic property of RNA. Okay, so do you have any questions? Any questions from anybody? Asiba, Anubu, Roshan, Fabina, Jency, Sibia. If you have any questions, any suggestions, you can tell me. Also, I noticed today that you had both of there are two TAs for this course, and you had both the TA classes at once. So, which is a little problematic. I mean, you cannot attend both the classes at once. So, if you want, you can ask the people. I will also try to contact them because it's not beneficial for you. You won't be, you will be the sufferer, right? Any problem, any question? Do you want, I just wanted to know that do you want me to take only the questions and end the class, or you want me to take the theory also? You want me to revise, just quickly revise the theory and to go to the questions, or only the questions? Because questions are the most important thing. This class is a question wala class. So just tell me. You can type in the message box whatever is your suggestion, because this class is for you. So I want your opinions on it. You want only questions, or you want theory plus questions? Please respond so that I have a better understanding. Hmm? How you want this class to be? Tell me. Asiba, how you want it? Anubu, Roshan, how you want this class? Theory plus question. Okay. So Roshan has said that theory plus question. Is this the same for everybody? See, you are the one who are going to get benefit if you tell me how you want it. Okay. I can go like others and I can just take questions, solve you 10 questions, end the class before 2 hours gets complete and go. But I'm not doing it just for your good, because I know that you have your coursework classes also, like whatever course you are in, and just going through the lectures, you might miss some topics or points. So I just try to explain you in brief in the class. Anyways, we have 2 hours time. We can cover theory and questions both. But it completely depends upon your interest. If you want that much, then only I do, because I am taking extra effort from my side to make you understand the concept. That is not my part actually. I just had to take those questions and just leave, but I'm putting extra effort just for your benefit. So Roshan has answered that question theory plus question is better. What about the others? Is it fine with everybody? Theory plus questions? I don't know why you don't answer in the message box. Anyways, if you are not answering, I'll just consider it that theory plus question is fine. So I'll take it in this way. So next week again, Tuesday, 7 to 9, my class is fixed, 7 to 9 Tuesdays. Uh, we'll solve the theory and questions. I mean, we'll just quickly revise the theory and then we'll come to the questions of your week 3. And I mean, it would be very good that if you come on time. Today, I understand that you had other class also. That's why you couldn't, you might not be able to make up. But try to come on time so that we can have a good interactive session. I'm also a student. See, I'm also a PhD student. I'm not a like a professor or something. So it would be better if it's we try to make it an interactive class. I learn something from you. You will learn something from me. It will be an interactive class. We will discuss. If you are unable to understand some points, you can ask, you can discuss it. This is actually a very great platform for you. It's a very good platform where you can clear your doubts. You can clear your concepts. You have someone, uh, live, where you can interact and you can clear your doubts. So it's up to you how you utilize this opportunity. Fine. So okay, then I think it's we are done for the day, and we meet in the next class, Tuesday, 7 to 9. So bye, everybody. You may leave the meeting now.